Topic is transport across the cell membrane. Now, if we look at transport across the cell membrane, there are different forms and different ways that substances are transported across the cell membrane, which is composed of a phospholipid bilayer. And we'll be talking about the cell membrane in its own topic in the very near future. But for now, I need you to understand that you do remember that the intracellular fluid compartment and extracellular fluid compartment are different from each other. If we just go back in our notes over here, we could see that the composition of the ICF and the ECF are quite different over here. So if we look at this, how is this created over here? Why is the composition different? The reason is because your cells and the organelles inside your cells, like the nucleus, the ER, and all these other stacked organelles, Golgi apparatus, lysosomes, vesicles, everything you name it, the mitochondria over there. I'm just drawing the typical appearance. This is the inside of the mitochondria. Anyway, so they need certain conditions to be met in their immediate surrounding environment to function properly. So for the biological functions that take place in the cell, you need to have the right composition of stuff in the cell. And when we look at the ECF and the ICF, again, there are certain processes that happen at the cell membrane. I'd like to actually say that a lot of physiology happens at the membranes. So if we look at this, many processes like the transport of action potential, that is how cells communicate with each other. Electrical signals are transmitted from excitable cells like your neurons and your skeletal muscle and cardiac muscle and some smooth muscle and even pacemaker cells in the form of electric currents that are generated on the cell membrane. So how is that taking place? Well, it's because there are differences in the ionic concentrations, and there are certain stimuli and certain conditions which, when met, would result in an action potential to be generated on the cell membrane and transmitted across its entire surface. And so for this, we must discuss about how these substances are transported across the membrane. And it's not always creating differences that transport across the cell membrane does, but sometimes you can also dissipate gradients or differences. This happens in the form of passive transport. So let's just classify transport across the membrane. There are two types under this. So transport can be passive or active. When I say passive, you should think of something that does not involve energy. No energy expended. No energy no energy used up. When we say energy, I mean ATP. And so what happens is there is a certain gradient, a difference between two compartments. There's more on one side and less on the other side. And what happens is the substances move from an area of higher concentration to an area of lower concentration. So it is going along its gradient, and it's not just concentration gradient, but sometimes even electrical gradient. Ionic charges can be different and can favor the movement of certain charges downstream again. So it goes along the electrochemical gradient. Electrochemical gradient. And if we look at active, here we have the use of energy. So we will need energy. So ATP would be expended. ATP is used up. Now it could be direct or indirectly. What do I mean? So in active transport, we have these things called pumps and transporters, and they are special protein structures on the cell membrane that allow the substance which we want to transport to bind to it, and then it flips along the cell membrane. So it transports this molecule from one side to another. I'll be talking about that in more detail soon. So when we look at this, we are using up ATP, and this means that we are able to push things against the gradient. So you can actually concentrate the substance on one side of the membrane. Even though there's already so much of it on one side, you can still add even more. You can extract it from one side of the cell membrane and push it to the other side of the membrane, which already has so much more. So the ATP is used up in active transport. Now, whether it is direct or indirect, if the pump or the carrier transporter directly binds to an ATP molecule, breaks down the ATP molecule and uses it for energy, then that is primary active transport. But if the carrier or the transporter does not bind to that ATP molecule, but instead uses it indirectly, through other mechanisms which we'll be discussing, that is secondary active transport. So here, because you have a pump, you are technically able to push things against their electrochemical gradient. So you're going against the electrochemical gradient. This is very important. Alright, so now when we talk about passive transport, the first heading I'm going to talk about is simple diffusion. I'll be dealing with passive transport first. So simple diffusion is where you look at molecules, and let's just say that hypothetically there are two compartments over here, and there is a certain substance. It could be gas, it could be fluid, whatever. Okay, so I have a few molecules in compartment A and very few of these molecules or atoms in compartment B, just a few. Now look, this is a fluid. Our body, like I said, is made of water, so it's a fluid system. So if we look at this, these fluid molecules are in random Brownian motion. Random Brownian motion. That is to say, they're moving haphazardly in all directions over here. Some will move this way, some will move to the right, up, down, left, right, whatever. So we have all these molecules moving left and right. And what happens is, this happens in both the compartments. Now, what happens is, if we look at this, the molecules that can move from compartment A towards compartment B, because this is that kind of membrane. So this separating barrier between compartment A and B has allows molecules to move across it, or from area A to area B. So now look at this. The molecules can they're moving across left and right, up and down, in any direction in three-dimensional space. And if you look at it, there are more molecules in compartment A than in compartment B. So what's going to happen? Some of these molecules are going to move towards the membrane and eventually make it across. You see that? And some of them won't. Now, there are some molecules in compartment B that are likely to hit the membrane and go to the other side. But if you look at it, the net movement is more from compartment A to compartment B. Why is that? Because there are more molecules in compartment A than compartment B. That is to say, the concentration gradient is the most important factor for determining the transport of atoms from one side to another, or molecules from one side to another over here. So this is like I said, through random Brownian motion. The best example I can give you is if you are in one corner of a room and you open a box of pizza, and the smell wafts over the entire room, and eventually everybody does smell that somebody did order pizza. So it can't be hidden, and that's because of random Brownian motion. And the stronger the spices and the flavor of the cheese and the oregano and the pizza, the much faster will the smell dissipate, will those flavor molecules dissipate in space, and everybody will eventually know that there is somebody eating pizza, like that. So now, can we calculate the amount of flux through simple diffusion? Yes, and we have mathematical formulas for that. So the first formula I'm going to tell you is Fick's law. Fick's law, F-I-C-K-S, Fick's law. So Fick's law tells me that the flux, the amount of substance that is transported across the membrane, you could easily calculate it with this formula. J represents flux. flux, okay, meaning the amount of substance moving per surface area of the membrane per unit time, is equal to, let's remember, what is this thing dependent on? What does this diffusion, simple diffusion, depend on? What makes it more? That goes in the numerator, and what makes it less goes in the denominator. So let's first of all add a constant. So we'll make it a minus sign. We add the minus sign in this formula. This is because the substances are moving from higher to lower concentrations, so that's why there's a negative sign on that. Then we have a constant called D. D is a constant, and this is your what we call the diffusion coefficient. Diffusion coefficient. This is from Graham's law. I'm going to talk to you about that in some time. So diffusion coefficient, and what else is important over here? Into surface area of the membrane across which this diffusion is taking place. So this membrane, the bigger the membrane is between these two compartments, the more chance it is that some molecule is going to hit that membrane and eventually cross that membrane. So this is where we have surface area again. The more surface area you have, the better is the diffusion. So imagine if you have a condition where your where the cell membranes or the surface area, the number of cells across which a certain substance must transport across is less, then what would happen? There would be lesser diffusion. Good example I can give you is emphysema. So emphysema is a subtype of chronic obstructive lung disease, and what happens is, in that, somebody smokes a lot, or they might have a genetic abnormality, they might have a deficiency in an enzyme. But what happens is, eventually, through smoking or a genetic defect, the respiratory tract gets destroyed, gets damaged over there, and this happens over a period of years if you're a smoker, or at a young age if you are quite if you are having the genetic abnormality, the genetic variant. It's an alpha-1 antitrypsin deficiency that can cause this. Anyway, so what happens is, in emphysema, because of the let's talk about the chronic smoker type example, because this is more common. So here, these chronic smokers smoke a lot, and they smoke for years and years, lots of cigarettes, lots of packs a day, and with time what happens is the airways get weak and damaged and destroyed, and the alveoli also start to get destroyed, and they don't function. So at least half of their alveoli don't work. If half of the alveoli don't work, they don't oxygenate so well, they don't ventilate their lungs so well, so the oxygen does not go into the blood that easily, and the CO2 does not go back into the lungs that easily. So that could be a problem. So surface area is very important. Times concentration gradient. This is very important. I told you, one of the important things that determine transport across a membrane. Now, in some cases, if we're talking about gas, like in the previous example I gave you about emphysema and oxygen and CO2, we can, instead of delta C, we can use delta P, meaning the partial pressure of that gas across the membrane. So imagine one side has more oxygen than the other. Like when you inspire air, you have plenty of oxygen in the inhaled air. So your alveolus gets a partial pressure of oxygen of 100, almost 100. It could be 103. And in your blood, if you see the venous blood that's coming to your alveolus, that partial pressure of oxygen is 40. So when that blood reaches the alveolus, then an exchange of gas takes place, and there's more oxygen coming into the blood. So then eventually you get a partial pressure of 100 millimeters mercury in the arterial blood, in the pulmonary venous blood that goes back to the heart and then gets pumped to the arteries via the aorta. So you could see that this is an example of diffusion over here taking place. And here we can substitute delta C with delta P, because the more the number of molecules, the more is the partial pressure of that molecule in that system. And it is inversely proportional to the thickness of the membrane. So you can add X represents thickness. I'm going to write this X like this. A little different over here. Don't mistake it for the multiplication signs in the numerator over there. So This represents thickness of the membrane across which the diffusion is taking place. Remember, it's all common sense. The thicker the membrane, the harder it is for substances to go across it. Let me give you another example. So another respiratory condition, because when we talk about simple diffusion, most likely you'll be thinking about the lungs, where gases get exchanged across the alveolar, across the respiratory membrane. So what happens is in your lungs you have your alveoli and you have the vessels. Now there is epithelium on the alveoli and there is endothelium in the blood vessels, and if you look at this connective tissue, there's a small layer of connective tissue between the alveoli and the blood vessels. Now what happens is there's a condition called fibrosis of the lungs. So in fibrosis, there are many reasons you can get fibrosis. It's technically scarring, like when you injure a part of your body and you get a scar and that scar looks ugly and thick. That's because of fibrosis. There's a deposition of excess amount of collagen. And this collagen that deposits, or this connective tissue that deposits, makes the distance across which your oxygen molecules have to move much higher. It increases that distance, so that makes it less likely for the molecules to go across the membrane or to make it across the membrane. So the thicker the membrane, the less is the flux that's taking place, or the diffusion that's taking place. So this is your Fick's law and its various applications over here. So the best examples are oxygen and CO2 transport in the alveoli. And the case of CO2, I want you to remember that the PCO2 in the partial pressure of CO2 in the alveoli is 40 millimeters mercury. In the pulmonary arterial blood, it is PCO2 of 45 millimeters mercury, mmHg, because it's partial pressure. Its pressure is over here, millimeters mercury. And by the time we reach this blood reaches the alveolus, that excess amount of CO2 will go into the alveolus, and now you are left with a PCO2 of 40 millimeters mercury. And this is how it should be. We need CO2 in our biological systems for life. They help in regulating the pH and so on and so forth. They have many important functions, especially pH regulation. So then after that we have the other heading, which is facilitated diffusion. Under passive transport, facilitated diffusion. Now, what is facilitated diffusion? Here, again, this is passive. That means there's no energy required, no ATP required, no energy required. You can use them interchangeably here. And you are, again, going along the electrochemical gradient.